Hey guys, welcome back to my channel. In this under 10 minutes video, I'm going to be going over IB Biology topic 1.2, the ultrastructure of cells. First thing we need to note is the difference between prokaryotic and eukaryotic cells. Eukaryotic means after nucleus, or after karyote. Prokaryotic means before nucleus. So these don't have a nucleus, these have a nucleus. So let's just write that down first. And instead of the nucleus, each cell has genetic material. Prokaryotes have it in this thing called the nucleoid region. And going on this notion, the main difference between prokaryotes and eukaryotes is that prokaryotes have these things called membrane-bound organelles. So that means that on top of the cell membrane, each of the organelles, or some of the organelles, have their own membranes. Like the nucleus has its own nuclear membrane, mitochondria has mitochondrial membrane. And what we call that is something called compartmentalization. That means, as you can imagine, divided into compartments. That helps in maintaining conditions that are ideal for certain parts of the cell. Like storing DNA would have different conditions that are ideal from, say, creating proteins, such as in the ribosomes, or actually a better example would be the mitochondria with cellular respiration. And the main features of cells include the cytoplasm, which is in both cells, prokaryotic and eukaryotic. Nucleoid is in prokaryotes. Plasmids are in prokaryotes. Plasmid is an independent circle of DNA, so it's quite related to this. And ribosomes are also in both, and based on the type of cell, they can be different size. So ribosome in eukaryotic is 80 Stargardt Swedberg units, and ribosome in prokaryotes is 70 Swedberg units. Cell membranes in both. That just is the phospholipid layer that surrounds the cell and contains all its material. Cell walls are in prokaryotic cells, and in plant cells that are eukaryotic. What that does is provide sturdy support for the cell and contains it. There's this thing called a slime capsule, which is sort of like a sugar layer that's used for protection of prokaryotes, and this is kind of called the peptidoglycan layer, I believe. Peptidoglycan layer. And that's sort of like a capsule that protects it from phagocytotic desiccation and breakdown. So flagella is also a component of prokaryotes. So flagella are these elongated projections and with motor proteins that allow them to sort of move around. So prokaryotes need this. Pili are also these things that help prokaryotes sort of attach to other prokaryotes and exchange DNA. So these are like hairs on the cell. So eukaryotes and prokaryotes are different in the sense that cell membranes, they both have cell membranes, but this one has compartmentalized membrane-bound organelles. And when we talk about prokaryotes, the way they divide is something called binary fission. And the binary fission involves the process of the nucleoid DNA first being copied, and next it's going to migrate to opposite sides of the cell, and the cell is going to elongate and pinch off, forming two different cells with the same identical stuff as the first initial cell, so 1 to 2. And that's pretty cool. That's a form of asexual reproduction. Eukaryotic cells include many types of cells that we know, such as protists, which are some of the bad stuff in the world, and you have fungi, you have plants, and animals. Of course, animals, us, plants, say a fern, and those all have these properties. Plants and animal cells, they're quite different, so these ones have different features that I'm going to go into. And organelles are the parts of the cells that are inside the cells itself. So cells have organelles in the form of like the ribosome, cytoplasm, cell membrane. We all consider those as organelles. Well, maybe not the cell membrane, but anyways, organelles. With organelles that are common to all eukaryotes and prokaryotes, we went over the ribosomes, cytoplasm, and plasma membrane. But the eukaryotic cells also have universal organelles. So the nucleus is one of these. All prokaryotic cells have a nucleus, whether it be plant or animal, and this contains the DNA and conducts DNA replication in getting ready for cell division. There's the endoplasmic reticulum, which is sort of like this transport network made of filaments, and it can be smooth or rough. And rough means that it has these things called ribosomes, which synthesize proteins. And there's the Golgi apparatus, which is sort of like a postal delivery service, which packages molecules and materials before it's sent out of the cell. There's the mitochondria, which is the structure which conducts aerobic cell respiration and makes ATP. There's this thing called the peroxisome, which I don't know. I believe it's not that. It's a bit more complicated, and it's a thing that contains lots of breaking down enzymes. So it's sort of like a lysosome you may have heard of, and but it's conducted with breaking down toxic materials. Next, we have the centrosomes, which are these structures that have microtubules that go out of it and are useful in cellular division. And these are actually in pairs in animal cells, and they're in single and by themselves in plant cells. Moving on to organelles that are only within plants, we have the chloroplasts, which are the sites of photosynthesis, and they contain stacks of these things called thylakoids, which contain the materials and chemicals for photosynthesis. Photosynthesis you'll learn about later. These things called the vacuoles, which store water. It's like a large vacuole. So animal cells have small vacuoles, but what plant has is this large central vacuole that just covers up most of it. So instead of like nucleus would be like this in comparison, this would be the vacuole that contains water and exerts pressure on the cell, creating something called turgor pressure, which will be important in topic 8 when we learn about plants. And of course, plants have the cell wall, which provides a sturdy structure, which is why trees aren't squishy, plants aren't squishy. And moving on from that, you have animal-only parts, which are the lysosome. And lysosomes are sort of like these vesicles that have lots of enzymes that are hydrolytic, so they'll break things down. And that's quite useful in detoxifying substances, similar to the peroxisome. Component of this topic regards microscopes, and specifically comparing electron microscopes with light microscopes. Now, electron microscopes can zoom in a lot closer, and they have a higher resolution. So resolution means that it can differentiate smaller things. So two smaller points can be differentiated by an electron microscope compared to a light microscope. So the two main types of electron microscopes are scanning tunneling microscope and scanning electron microscope and transmission electron microscope. And the scanning electron microscope is, as I called it tunneling, was because it sort of has like you can measure depth of things and how they go down and stuff, and measure the surface, so it's kind of like a terrain. And transmission is that it's the most zoomed in, so you can zoom in a lot. And the disadvantage with electron microscopes compared to light microscopes is that, of course, you can't see different colors as with light microscopes, which is why you have to stain these ones with a particular color, so that you get those, be able to differentiate substances or materials or sections of the cell. And that's why you see weird images with different colors that shouldn't be there. Building on from this are these things called cell micrographs, which are basically these black and white images of cells and different parts that the IB will get you to label and recognize parts of. So those are quite important for tests. I recommend just practicing. Next, you're going to have to know how to label a prokaryotic cell. So the main components are the nucleoid region, the cell membrane, the cell wall, the pili, and the flagella. Also, you have to label the 70S ribosomes. Make sure you say 70S, and also the plasmid. So those are key features that will give you. What you need to know how to draw is the eukaryotic cells, the plant and the animal and the plant cell. So this is animal, this is plant, and each of these will have the nucleus and the nuclear membranes right here, and the nucleolus. Make sure you label that separately. Both have mitochondria because they both have cellular respiration. That's aerobic. So both need mitochondria. You also have ADS ribosomes in both of them. Both of them have a cell membrane. Plant cells have only cell wall. They have also a cell wall, and these have a central vacuole. That's important to draw. And both of them have rough endoplasmic reticulum, so rough ER, and they both have smooth ER. Animal cells also have the lysosome, and the plant cells have the all famous chloroplast, which is important in photosynthesis. And that's it for IB Biology topic 1.2. See you guys in the next video, 1.3, phospholipid bilayer.